purpose of this video today is to show exactly how nucleic acid point mutations can affect the sequencing of DNA and the amino acids that are produced. Here we see a sequence of normal DNA that I'll use throughout the video to demonstrate missense, nonsense, silent, and read-through point mutations. It would be very repetitive if I read off every single base pair, so moving forward I'll point out the base pairs that have been mutated and show the effect that this will have on the amino acids that are produced. So if this DNA does not have a mutation that occurs, the amino acids that would be produced from this segment of mRNA would be methionine, tryptophan, asparagine, alanine, cysteine, and then the UGA base pair would code for the stop codon. If there were a missense mutation that occurred, like it is provided here in red, where the cytosine changed to a guanine, which makes this a transversion mutation, the cysteine amino acid would now be coded for another tryptophan amino acid. This may or may not cause either a loss of function or gain of function down the line, as mutations are accountable for changes in protein function. In this next example, we see there is a mutation of the same cytosine base pair, except this time it's mutated to an adenine. This is also an example of a transversion mutation, as it is changed from a pyrimidine to a purine. However, it would be considered a nonsense point mutation because it changed the location of the stop codon where it was originally a cysteine amino acid. The base pairs that have been mutated and the effect that this has as it undergoes transcription and translation is again shown here in red. Here we see in red that a transition mutation has occurred as the cytosine has mutated into a thymine. However, if we look at the amino acids at the bottom, we can see that there has not been a change from the original sequence, despite there being an alteration to the mRNA portion that codes for cysteine. The original base pairs on the mRNA, UGC, has changed to UGU, but this sequence of base pairs still codes for the amino acid cysteine. This is an example of a silent point mutation. Lastly, here we see an example of a transition mutation as the original base pair on the coding strand has mutated from an adenine to a guanine. As this mutation undergoes transcription and translation, we see that the codon that was originally coded as a stop codon has been changed to a tryptophan amino acid, which means that the protein will now be much longer. In this example, after tryptophan comes a proline amino acid, followed by a glycine amino acid, and the red dots following this show that the polypeptide chain will continue on until it reaches another stop codon. This is what we call a read-through mutation. Again, as with all the previous mutations, we can see that these may alter the functioning of the proteins. In some cases, however, as we saw with the silent mutation example, this may not be the case, as the amino acids may not actually change.